Welcome back to that. We're going to start our screencast over cells. You guys are in for a treat tonight. I'm using a Prezi which I've made before and before time, so I hope you enjoy this Prezi and this screencast. So first of all, when we get started, when we talk about cells, there are two main types of cells that we need to learn about. The first one, those are prokaryotic cells and eukaryotic cells. The first one, prokaryotic, if you remember, the word pro means before and kary means nucleus, so this means before nucleus. So we're talking about cells that don't have a nucleus. So if we look at the actual definition of a prokaryotic cell, it do not contain membrane-bound organelles such as the nucleus, the mitochondria or chloroplast, or any other organelles. So we usually think of it as just the nucleus, but remember there are other organelles inside that cell that they don't contain either. A good example of a prokaryotic cell is a bacteria. Remember, so we're all familiar with bacteria. They're much smaller, aside from not having the membrane-bound organelles, they are much smaller than eukaryotic cells as well. So when we look back at this image of this prokaryotic cell, you'll notice that some features that these have that eukaryotic cells have as well. There's a cytoplasm in there. They both have ribosomes. The plasma membrane, both eukaryotic cells and prokaryotic cells have a plasma membrane, and they both have DNA in the middle of it here. But you'll notice there is no nucleus. They just simply have what's called the nuclear area or the nucleoid region. The one thing that they will have in common too with some eukaryotic cells is this cell wall. Plant cells have a cell wall, so do prokaryotic cells. So the next thing we want to talk about are eukaryotic cells, and most of our time is going to be focused on eukaryotic cells. The word eu meaning true, kary meaning nucleus, so it has a true nucleus. Easy way to remember it, eu are made up of eukaryotic cells. So you think of animal cells, plant cells, fungal cells, the protists, things like that. Those are all eukaryotic cells. And a definition would just be the opposite of the prokaryote. These eukaryotic cells actually have membrane-bound organelles. And let's kind of compare the two here, a little comparison and contrast. Prokaryotes have the nucleoid region. They also have a circular DNA, where eukaryotes have a nucleus, they have organelles, and they have a linear DNA. Now, things they have in common, they are both types of cells. They both have a cell membrane or the plasma membrane. They both have ribosomes, but also remember that some eukaryotes, like plants, have a cell wall. That would be something else they would both have in common. So let's go ahead and jump into this eukaryotic cell here. Before we get into that, I want to talk about overall cell size. In your book, there's a little section that talks about how it's important for cells to the reason that they're so small is that it allows them to be more efficient at getting nutrients into the parts of the cell that are needed and to get rid of waste. So if you look at this picture here, we have two cubes. Roughly, they have the same volume, 125 here. But if you'll notice, this one here has a much smaller surface area as opposed to this one, because this one is broken up into much smaller cubes. So what happens here is, as a cell, it can be a cell or any structure, as it increases in size, the volume is going to increase much faster than the surface area. So what happens is that gives you a low surface area to volume ratio. So one of the things that cells want to have, they will have a high surface area to volume ratio, and that allows them again to be more efficient in taking nutrients into their cells and getting rid of wastes. One thing to remember is that just because, if you have really large animals, so if you think of a blue whale is not going to have really big cells because it's so big. Blue whales are going to have just more of the regular size cells, the same size cells that we have. They're just going to have more of them. So that's just a little bit about that importance of the cell size. So the first structure we want to talk about is the plasma membrane or the cell membrane. You should be a little bit familiar with it from the last chapter because it's composed of that phospholipid bilayer. And remember, the cell membrane acts as this barrier that keeps the outside of the cell from the inside of the cell. So it's a selective barrier that keeps certain things out and allows certain things into the cell. So we'll take a closer look at it here. You'll see the main component of the cell membrane is that phospholipid bilayer. And you'll also notice these large purple structures here in this drawing those are proteins, and their jobs are to allow certain objects and certain things into the cell that can't freely diffuse through the membrane. These little structures here, these cholesterol molecules, are embedded throughout the membrane, and their job is to help that cell membrane maintain the proper fluidity. Remember, cell membranes are fluid. They're oftentimes called a fluid mosaic. So they move around a little bit. There's some fluidity to it, and those cholesterol molecules keep them at the proper fluidity so that they don't get so fluid that they just fall apart, and you don't want them to be so static that they don't move. So then they're rigid. So that those cholesterol molecules help maintain the proper level of fluidity in that cell membrane. We'll talk a whole lot more about the cell membrane in the next chapter. The whole chapter is focused on cell membrane and the transport of molecules into and out of the cell, and we'll learn all about that then. The next structure we're going to talk about is the nucleus. Of course, we want to think of the nucleus as like the brain of the cell because it contains information, just like your brain contains information, hopefully. So within the cell, you'll notice that the nucleus is going to house your DNA, which is oftentimes organized into chromosomes, and that's the structures that's going to carry all your genetic information. And so we keep that inside that nucleus to protect it. It just gives it that extra protection inside the nucleus. Within the nucleus, we have a structure called the nucleolus. And the nucleolus, there can be more than one, in which case we call it the nucleoli if there's more than one nucleolus. But this is where ribosomal RNA is made. And that's what that little R stands for in front of that RNA, is ribosomal RNA. And that is a building block for ribosomes. And the ribosomes are going to be made outside the cell. But the building blocks for all those ribosomes are made within the nucleolus, and then the ribosomes are going to be found outside, which we're getting ready to talk about here pretty soon. The next structure that we want to get to is the ribosome, and I have a little short video. One thing I want to mention before we get to the video is talking about ribosomes is you'll notice these little brown gold dots throughout this. These are the ribosomes. Now, they can be found in two forms. We have free ribosomes, which are just found in the cytoplasm, and then we have bound ribosomes, which are going to be found attached to the endoplasmic reticulum, more specifically the rough endoplasmic reticulum, and we'll talk more about that in a little bit. But I have a little short video. We're not going to watch very much of it here, but if you'll, it talks a little bit about ribosomes. We can get it to work here.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules.
We're going to stop that there. So hopefully you saw where the main thing we wanted to get from this video is that the little purple structure here is a ribosome, and the strand here that you see is RNA. We'll learn a lot more about this later, but it's feeding through this ribosome and it's going to be making proteins. So the key feature to a ribosome is that it is the site of protein synthesis. Pretty easy to remember, hopefully. And so that is what a ribosome does. It's the site of protein synthesis. The next structure we want to talk about is the smooth endoplasmic reticulum. It's one of my favorites. It's pretty cool the way it works. First of all, you'll notice we call it the smooth ER because it doesn't have ribosomes attached to it. It has a couple of jobs. First of all, it's a place where lipids, which include steroids, are going to be made. But one of the main jobs that I want to focus more on is this ability of the smooth ER to detoxify drugs, toxins, poisons that get into your blood. They're usually especially abundant in your liver cells. Now, one of the things they do in your liver cells is that they're going to be there, and that's the job of your liver, is to filter your blood and to take the toxins out of it. Cool thing about the smooth ER, within the liver cells, when people are abusers of drugs or alcohol, their liver cells will try to accommodate that, and they will start to proliferate, make a bunch of these smooth ER organelles throughout the liver cells. And that is specifically so that they can counter all the drugs and toxins that are coming into their body. So in turn, what ends up happening to these alcoholics and these drug abusers is they have to take more and more of the drug or the alcohol to get that same effect that they're looking for, because their body's just producing more of the smooth ER within their liver. And eventually, you know, what happens is that liver will just wear out, and cirrhosis might kick in, and you have liver failure, and you can just overwork your liver is what happens with that when you start producing so much of that smooth ER. So that's a little bit about the smooth ER. The other type of endoplasmic reticulum is the rough endoplasmic reticulum. You'll notice the rough ER has ribosomes embedded throughout the folds of its membranes. And of course, because it has the ribosomes attached to it, there's some other functions of the rough ER, but one of the main functions we want to focus on is that it is also a site of protein synthesis. So again, so that's a little bit about the rough ER and the smooth ER. That's a good place to stop tonight. We'll continue on with the next video. We'll pick up with the Golgi apparatus. So hopefully you guys keep reading your book, stay ahead of what we're studying, and it will help you immensely. I'll see you later. Bye.